When I started to work on microglia cells, we were really exotic people because almost nobody was working on microglia cells. Most neuroscience people were working on neurons, and microglia cells were really considered as very boring.
Microglia are the brain's garbage collectors. Studying them for a lifetime probably sounds boring to most people. However, for almost 25 years, the research of Marco Prinz and colleagues has revealed that microglia are so much more. Today, many companies are trying to target the cells therapeutically to treat serious brain disorders. For his pioneering studies of microglia, Marco Prinz is receiving the 2021 Novo Nordisk Prize. After completing his medical studies in Berlin in 1996, microglia cells were not at the top of Marco Prinz's wish list, and the opportunities for research in the former East Germany were very limited.
We use silver impregnation techniques that were from Cajal more than 100 years ago. This gave us wonderful pictures of neurons, but nothing more. We couldn't afford to buy antibodies, for example, to have more insights. But this was a starting point for me to get interested in the brain.
In an attempt to integrate East and West after the fall of the Berlin Wall, new research institutes were built. One was the Max Delbruck Center for Molecular Medicine in Berlin, where Marco Prinz looked for opportunities.
One group was working on stroke, another was working on brain inflammation, meningitis. And actually, the stroke group was full, and that's why I had no choice. I had to join the other group.
Whereas most brain researchers worked with neurons, biochemist Uwe Carsten Hanisch studied a special type of cells, microglia.
People considered microglia as very boring cells. If a party is over in the brain, microglia come into the play and remove all damaged cells from the brain to clean it up. The brain is separated from the rest of the body by the blood-brain barrier. That means you have no influx of cells, of circulating cells, coming from the blood to the brain.
The brain therefore hosts its own immune system, in which microglia cells play a key role. However, in 1997, brain researchers showed little interest because their focus was elsewhere.
I was looking around, okay, what is the best option to continue in research and still having connection to patients? And at this time point, the prions, late 90s, was quite popular. So in the United Kingdom, thousands of kettles got this BSA, and in Europe there was a big crisis and people were concerned about new variant of Creutzfeldt-Jakob disease.
A world-leading prion expert, Adriano Aguzzi from the University Hospital of Zurich, took Marco Prinz in as a postdoc.
In the beginning, I was quite shocked to be there because to be exposed to brilliant people coming from different countries with different backgrounds, and I was more or less a newcomer. The neighboring department was headed by Ulf Zinkernagel, Nobel Prize winner in immunology, and we had joint meetings with Charles Weisman, who cloned as the first time type 1 interferons. I got exposed to this environment, how to do research, how to sell stories, how to write grants, and this was extremely helpful for my future career.
After publishing in major journals such as PNAS and Nature, describing a mechanism for the transfer of prions to the nervous system, Marco Prinz became group leader at the University of Göttingen. But convincing potential donors and research councils that studying inflammation of the brain is important was difficult.
I wrote several applications to get fundings, and most of them were rejected just by saying, okay, he did wonderful work in the past, but it was on prions and nothing what he wants to do in the future. And it was quite frustrating.
However, almost overnight, Marco Prinz's luck changed. And the start of the 21st century turned out to be the pivotal time for neuroscience.
There was a revolution in medical science. New microscopes were developed, new transgenic techniques were available, and this suddenly put new light on microglia cells.
Using green fluorescent proteins as labels, researchers could now remove existing microglia cells and replace them with donor bone marrow cells to see what happened.
This was really a breakthrough because we could show that maybe exhausted in the older brain can be renewed and repopulated and refreshed.
Studies of patients that lack functional microglia cells made researchers in the field realize that microglia are not just boring garbage collectors.
These patients can develop Alzheimer-like or Parkinson-like pathology. This was the evidence that microglia cells, even in the healthy brain, are important to feed neurons and for establishing the homeostasis in the brain. That they are kind of gardener in the brain. They are sitting in close vicinity to neurons.
In 2008, Marco Prinz became professor at the University of Freiburg and responsible for neuropathological diagnostics at the university's medical center. In an exciting study in 2015, Marco Prinz and colleagues made a pivotal discovery when they investigated germ-free mice without any microorganisms living in the gut.
We recognized quickly that microglia cells in these animals are totally disturbed. We also identified the molecules derived from host microbiota to the brain, and we could identify so-called short-chain fatty acids that are produced by gut microbiota, signal to the brain. This was totally surprising to see that the gut, our host microbiota, continuously speak with the brain and continuously educate the microglia cells in the brain.
The study became the starting point for experiments in many other laboratories.
In some patients, for example, multiple sclerosis patient, the microbiome is different. What is not clear at the moment whether this is causal or secondary, and whether any manipulation of the microbiome could lead to changes of the CNS and CNS diseases.
It was soon clear that microglia are indeed drivers of many diseases of the brain.
Researchers performed in thousands of patients suffering from Alzheimer's, from Parkinson's, from multiple sclerosis, genome-wide association studies, and we're looking for the risk factors. And it's now clear that all of these diseases have hundreds of different risk factors, and the majority of the risk factors for these diseases can be found in microglia cells.
Marco Prinz and colleagues had to develop new and innovative techniques.
But it was not clear how this network of microglia is established and how this network will be changed during pathology. And to overcome these problems, we generated so-called confetti multicolor microglia cell lines in mouse that allows us to label microglia in different colors. If you have a kind of pathology, these cells rapidly activated and clonally expand.
Today, the exploration of what were once the most boring cells in the brain has revealed that microglia are much more than just garbage collectors.
Microglia cells have to be multitasking cells. Yeah? That means they have to live for long, to keep memories, they have to react upon any stimulus, any changes of the environment. This might be bacterial infection, viral infection, but also if a neuron is dying, they try to help the neuron surviving.
Neurons are naturally the key cells in the brain, sending signals and information and creating long-term memories, but they need the microglia gardener cells.
Microglia cells are important to sense the signals of neurons, and they can actively remove synapses for neurons. Microglia cells are the important gardeners that cut the supernumerous synapses that we have at the end of the day a nice neural network.
Furthermore, microglia are also very sensitive and can respond or reverse in just minutes.
They are now considered as alarm clocks in the brain that are able to sense any changes of the environment very quickly, even before neurons and astrocytes get the information. And I hope that in the future we were able to use this feature of microglia as alarm clocks to label microglia cells and use them as early sensors of changes of the brain pathology.